Good morning. We welcome everyone to the webinar, Beneficiaries and Opportunities on the Use of Biol. Thank you for connecting right now. I will share what we will be seeing. I will share my screen with you now. As a first point, we have the registration of the participants is what we were doing previously. And the next point is the opening words. Then the presentation of the results of the project, the comments of the scientific committee. We will have a space for questions and answers. Then a panel of discussion on the conclusions and recommendations. And finally, the closing words. We remind you that at the bottom bar of your Zoom window, you have a Q&A button for you to ask your questions in accordance as they arise. You don't have to wait for the Q&A section. You can go ahead and write them down now, although they will be responded for the section, the slot that we have assigned to respond to these. You can utilize the chat if you want to make any kind of contribution or comment where you are connected today. And I see some comments of people who are greeting. Thank you very much. And we remind you that the project of investigation is part of the state of knowledge and use of the bioprovisions of the Andes, coordinated by Rikolto and the University of Michigan with the support of the McKnight Foundation. As a first point, we have the opening words. These is Vanesa Ramos will be giving us these opening words. She's a coordinator of the feeding systems in cities in Peru. So we welcome you. Good morning to everyone. My name is Vanesa Ramos, coordinator of the food systems, sustainable food systems in Rikolto. In the name of the organizers of this event, I welcome you to this virtual seminar that benefits, myths, and opportunities on the use of biol. It's a pleasure for us to have your participation today during this meeting, which is part of the investigation project, as Marcia previously mentioned. Knowledge and use of the provisions in the Andes, which was developed by the University of Michigan, Rikolto, with the support of the McKnight Foundation. The objective of this webinar is to deliberate the main results of the investigation and to generate a space and debate of bio. Likewise, this seminar seeks to visualize and to help to demystify some impacts and use attributed to this provision that had not been proven. This investigation has been developed under the umbrella of the collaboration program for resilient food systems of the McKnight system and adopted a holistic focus and ecosystemic using scientific knowledge with empirical knowledge of the family producers and rural producers in the Andean region of Peru, Bolivia, and Ecuador. We start to generate information of practical knowledge that is useful to have a greater productivity in cultivations and greater means of life for family cultivators. On the other hand, from the program of food systems, sustainable food systems of the cities of Rikolto, one of our priorities is to build evidence that foments more sustainable production systems that help us to have a better offer of fresh foods for the producer. And this is, we work building and strengthening alliances among diversity of actors, and we are interested in sending this information today to agriculture specialists, civil society, the academia, decision makers to have a greater impact of this study. So in this session, a little bit more of an hour and a half, we will have 4 moments. The first is to present the result of this study that has a structure of what we know and what we don't know yet of the bios, with emphasis on bios. Then we will hear the reactions of the panelists who are part of the scientific community who have accompanied this process. And then we will have a space to invite you to add comments and to ask questions and generate a discussion. And finally, the panelists and presenters will present their final comments. Once again, we thank you for your presence, your time today, and we hope that this seminar can be beneficial to you and fulfill with your expectations. Thank you. And I want to take advantage to remind you at the bottom bar, you have a button to activate interpretation in case that you would like to listen to this meeting in English. As a next point, we have the presentation of the results. This is in Vanesa Ramos will be presenting this, and of Brenda Uni from the Department of Science Appliance and Microbes of the University of Michigan. So we give them a space to know the results of their investigation. Okay. Thank you, Marcia, and thank you to everyone. Good day. Thank you to everyone for joining. Today, we are happy to share with you the results of an extensive revision of the literature and field magazines on the use of bioinputs, especially biol in the Andes. This work was initiated and supported by the McKnight Foundation. First, we will talk about on our findings, specifically the benefits, myths, and opportunities of the use of biol. The motivation of this work, the role of the bioinputs, in particular biol, as an agricultural practice in the high Andes. Our study has an objective to understand the current state on the knowledge of biol and the bioinputs. We carried out an extensive study of literature, reports, and scientific literature. We also conducted interviews in multiple regions of Peru, Bolivia, and Ecuador. Today, we will share results from this effort, starting with key definitions and background, participation and application of bioinputs, and finally, key findings, conclusions, and next steps. First, a literature revision. Including formal scientific studies in and outside of the Andes. We also focused on the university thesis, promotional manuals, and reports. We carried out interviews in 8 Andean provinces of Bolivia, Ecuador, and Peru. We connected scientific knowledge on the 2,000 meters above sea level. We synthesized our finding in a report that today we share. First, we clarified terms encountered in the review. Starting with the most general terms used to describe amendments to agronomic systems. A biostimulant can be either organic, such as compost tea, or inorganic, such as mineral. A bioinput is necessarily organic in origin, such as manure or plant extract. The term bioferment appears primarily in promotional literature, used to describe fermentations of organic ingredients. A mineral brew is a dilution or suspension containing ingredients such as ash, rock, dust, or salt, not fermented, which can be combined with any of these other inputs. The term biofertilizer is used widely and consistently. This refers specifically to preparations of microorganisms applied to plants. A biol is the most specific amendment and may be described by all of these terms. Specifically, a biol is a liquid product of an anaerobic fermentation of animal manure and other ingredients used as crop amendment. The use of liquid ferments in agriculture occurs worldwide. 3 factors contribute to the use of biol in the Andes. First was a promotion in 1970s in the Andes of digesters to produce biogas based on the animal manure and other residue. Liquid residue of this process was called biol and applied to crops. From the use of digesters developed in small batch fermentations, often called biopreparations, biofertilizers, bioferments, made in simple containers, prepared exclusively as crop amendments, and many also termed biol. Many of these fermentations have manure as the main component, with other ingredients varying widely, often adapted to local conditions and promoted through grassroots agroecological movements in Central and South America. A third factor, decades of careful research has demonstrated how some microorganisms directly benefit soil fertility and plant growth, making nutrients available, producing plant hormones, and suppressing diseases. Development of biofertilizers containing these specialized microbes has grown into a large commercial endeavor. A linkage is often explicitly made between plant growth promoting microbes and microbes that predominate in biol, creating a mystique that these fermentations contained special properties. Most fermented bioinputs, such as biol, are prepared in simple enclosed chambers such as sealed plastic drums to create anoxic conditions with a hose or valve for gas release. Our review found over 100 ingredients reported as additions to biol, including manure, 30 types of plant residue such as legume residue, herbs, and fruit waste. Other organics such as sugars, ash, and urine, various inoculums from commercial products to forest soil, and non-organic ingredients from rock dust to copper sulfate, and even synthetic fertilizers. Bovine manure is the most common input, added in the greatest amount, on average 25% of volume. According to interviews with agriculturists and promoters in the region, some ingredients are considered essential. Manure and unrefined sugar, for example, but the interviewees widely varied in what they considered other essential ingredients, for example, certain herbs, inoculums, fish guts, etc. They also noted the willingness or perceived necessity to purchase some inputs such as the container, or inputs such as milk or minerals. Regardless of the large variety of input, they are similar in containing ingredients such as energy, rich carbon components, many of which contain nutrients such as nitrogen, phosphorus, micronutrients at the start of fermentation. Literature reviews and interviews reveal practitioners allow fermentation for 20 to 90 days. Fermentation is driven by microbes consuming the energy-rich carbon material. During this process, carbon is released as a gas, mostly methane, and carbon dioxide, CO2. From 20 to 95% of the carbon can leave the bile this way, and the remainder is in the liquid bile or solid biosol. With the breakdown of organic material, nutrients are released. For example, some nitrogen may be lost as gas, but most will remain in the bile, a plant available from Anonymum. All the released phosphorus will remain in the bile, and much of it will be associated with solid biosol, solid bile. Other minerals, for example, zinc, magnesium, potassium, will be released into the bile and some associated with the biosol. The primary effect of this is a decomposition, accelerated decomposition of the atom material with such as in the bile and biosol. What else does bile contain? Our review found that the ferments of manure and plant residue also contain a wide variety of organic components. These include remains of original ingredients resistant to decay, such as tannins, terpenes, ligands. Bile also contains vitamins and organic acids, including some components which may promote plant health and growth. These components may derive from the original manure or plant residue. Microbes may also produce similar compounds during fermentation. The biomass of microbes may also remain after fermentation, together with the plant nutrients released into the bile. The final product contains a cocktail of these organic compounds. The amount of nutrients and type of compounds depends largely on the properties of the starting material. Together with a large variety of ingredients, our review found a large variation in how the final bile is applied to crops. Some field manuals and promotion material do not have recommendations for frequency or concentration of application. Our review found recommendations for applying bile to a concentration less than 1% and up to a third part of 33%, with a frequency of application from 2 to 12 times during a crop growth. Generally, the liquid bile is applied as a foliar spray, and it is rarely mentioned. It's used for specific crops. Now, the objective of this presentation is to share what we do know and what we do know of bile from the revision of the literature and from the perceptions of the producers. We have organized this part as questions and answers from the evidence and the conclusions of this study. If we ask if the bile increases or if it changes the biodiversity of the ground, we have seen that there are a few studies that speak of this issue, and there's no specific evidence that indicates that bile changes the composition of microorganisms in the liquid form. Can stimulate the activity of the bacteria that are present, but there's no evidence that this diversity increases in the species. So we can affirm that the organism of the ground is resistant to the change. This comes from study in general, and the investigations have demonstrated the microorganisms introduced in the ground do not last a long time. What can happen is that with practices of in a long term, the change of the composition of the diversity of the microorganisms. Regarding the application of bile, can applying bile increase soil carbon? There is no evidence. Scientific evidence to answer this question. We know that the fermentation process itself converts organic carbon into carbon dioxide and methane gas. We can affirm, yes, that the remaining biosalt solids that are in the base of the biol may contribute to soil carbon. If we ask, does biol contain abundant plant beneficial microbes with stimulant plant growth? Some studies show the presence of potentially plant beneficial microbes. Some indicate the contrary. In synthesis, we have no evidence that microbes in commercial inocules are predominant in the final biol. No study shows any direct benefit to plants from the microbes present in biol. We can affirm that the fermentation conditions used to make biol to produce this bio input favor the growth of the same microbes in the fabrication and manufacture of biol. It is possible that these microbes from biol may have some direct or indirect benefit in plants. It is difficult to distinguish if these factors that we can see in the plants are only from microorganisms or other factors, such as the effect of the macro and micronutrients. We have another question from the perceptions of the producers and the different testimonials that we collected. Do the sugars, such as molasses, which is a common in biol, stimulate microbes, the production of biol? There's no scientific evidence that adding sugars, such as melaza or molasses, promotes beneficial microbe production. There is no study that relates that adding molasses to nutrients helps with the nutrients to biol. We can affirm that the microbes grow with high amounts of sugar that change the pH of the biol, increasing its acidity. A low pH may preserve organic acids and soil ammonium that contains nitrogen in the final biol. Does biol contain plant-stimulating compounds? Yes, some studies detect plant hormones in biol. Some of these studies show that some biol do not have these compounds. This is due to a lot of recipes to develop biol. What can we affirm? Biol contains a large variety of compounds similar to plant hormones and other signaling molecules. It is unclear, however, to what extent these compounds contribute to plant health. Does biol increase plant yields? In perceptions, a great amount of producers of the 8 provinces involved in the interviews observed or confirmed that this was an impact of biol. From the side of the scientific revision, we have 10 studies of all those reviewed showed no effect of biol on crops. 16 studies show a benefit to crops compared to just adding water. 2 studies show a negative effect of biol on crops. And one drug comparison with chemical fertilizers showed higher crop yield with biol and compost added together. What can we affirm? That the studies in general indicated that there's a greater increase, a greater production, but these studies, at the same time as these are controlled studies, utilize a high concentration of biol and very frequent applications. Many times this is not true in regular cultivation. Yes, as in the germination. Does biol help plants recover from freezing or hailstorms? This is a recurring question in the higher areas of the Andes. There's no evidence in the literature. It's not an issue that has been frequently discussed, but has been reported in testimonials of cultivators. What we can affirm that bile has the properties of a foliar fertilizer and may have some plant stimulating compounds. It is highly probable that bile will help plants with physical damage, such as from freezing or even pest damage. And can bile act as an insecticide or fungicide? We have received many testimonials from producers that consider the bile as it's useful for everything. We have no clear evidence if bile functions as an insecticide or fungicide. What we do know that the biocidal properties may be difficult to assess given the high variability of bile ingredients. Finally, does bile have human pathogens? Bile can contain pathogens and also reduce pathogens. The fermentation can reduce these pathogens based on literature. What we can affirm from this study is the content of my pathology of bile is explained because the main ingredient in volume is manure of some type of farm animal, and it contains pathogens, but these pathogens are the final product of biome, don't have to do with any kind of fail or any kind of process. And what is the use of bile? Is the use of bile widespread in the Andes? From the interviews carried out, many farmers expressed awareness, and some of them knew of it and they didn't know it, of the sample that we had, and that they used it as an agricultural practice. However, regarding the efficiencies, they affirmed that there's difficulty to understand the process of fermentation that it is implied. There's some confusion in the preparation and the application. What we can affirm from this study is that bile is promoted as from different NGOs and circles that promote agroecology as a local resource for fertility with special priorities for plant growth, which appeal to farmers who feel that this can help them to have a better production for the market in spite of promotion, the ample promotion from different projects carried out by NGOs. We find that bile use is more specialized to a limited set of farmers. Why is bile not used by farmers? From the interviews with those who have carried out the formation and accompanied different producer groups, the 8 provinces where we interviewed different stakeholders, they mentioned a lack of knowledge. They don't understand what bile is, a fermentation, and what is useful. Other times, they say a resistance to changing and leaving the agrochemicals to use a different kind of input that can imply a longer time. On our part, we can affirm that the interviewees also promised a lot of enthusiasm. They used benefits, but they didn't see the benefits that they expected in the field. They used bile with this perspective of having a clean production, and this did not occur. Finally, others mentioned that there weren't good results in chakra, and this was due to an erroneous use, a bad mix of bile, and there was no knowledge of the correct time to utilize bile. Why do farmers in the Andes use bio, I mean the group of those who have adopted to the practice. This has to be related to connections to farmer groups with regular, ongoing training and support for agroecological practices in general, not just bio. Thanks to some group dedicated to the production of others, and they exchange experiences and correct this deficiency that can arise in some errors in the application, diffusion, and the frequencies of use. This helps to maintain the practice. From the study, we saw that there are different reasons for using bio. One is that they're interested in having healthy food production. Another is that they feel empowered through participation and with projects with NGOs. Another is because they reuse on-farm waste. Bio is a very cheap fertilizer. Likewise, due to the environmental benefits that they can see, the perception is always that the market will prefer agroecological products. Some conclusions and next steps. The main products of liquid manure-based ferments are plant macro and micronutrients released during fermentation. Diverse organic compounds such as plant hormones and secondary metabolites from plants and microbes also occur in bio, but are less understood and more difficult to access in terms of their impact on plants and soil. Unsubscribed claims of the benefits of bio risk propagating myths about the contributions of inoculants and may distort real benefits. The diversity of inputs used in liquid ferments create opportunities to adapt ingredients to local regions. But a challenge to standardize their use. Bio can stimulate the soil ecosystem, including increasing mineralization and soil respiration. Monitoring soil health metrics will help evaluate direct or indirect soil ecosystems effects from application of bioferments. This information will inform agronomic use such as application rates and adaptability to local soils and cultivators. The same is true of biosol, which has distinct properties such as high organic matter and phosphorus. A better counting of inputs and economics will help assess the role of bio. With access to a greater amount of training and experience and flexibility of tests and error to maximize the benefits of the cultivation, the techniques, the collective techniques, and the training that use partition methods can partially address this limitation. The optimization of the use of farm-made bio in agroeconomic systems may be different from that of bio generated by a biodigester. Through definition, is a subproduct of the biogas production. Here we list several priorities for advancing the research and application of farm-made bioferments. The complexity of the application seem to restrict the use of these liquid ferments. So we will analyze some priorities to progress in the investigation application of bioferments developed in Chakra. First, develop a user-friendly tool to estimate the nutrient contents for a biol-given basic inputs and its estimated impact on yield for a crop at a recommended application rate. This would help standardize the preparation of liquid ferments with various ingredients readily available in each local context, as well as its use on crops. Over time, with careful modification, such a tool could estimate the fraction of nutrients in the biol and biosol. Simple experiments may combine biol addition with equivalent additions of basic plant nutrients to assess added benefits of biol, such as secondary compounds, plant hormones, vitamins, etc. This should also be tested in terms of pest control or physical damage to plants, such as recovering from freezing. Finally, determining the effect of frequency and rate of biol application on plant traits for different crops and careful monitoring of the impact on plants, and to carefully monitor the impact in the health of the soil, biological and physical metrics. It is fundamental to evaluate this practice. Final considerations. Integration, agroecosystemic integration. This review indicates that biol performs best in conjunction with practices such as compost amendments, future and other practices of agroecology. Future research should account for how biol fits into broader approaches to agroecosystem management, resource use, and sustainable livelihoods. For example, manure and plant residues are fundamental for the fertility management in the Andes, and diverting these resources to produce biol may not be beneficial for some producers. Biol should be developed in the context of sustaining agrobiodiversity in communities of the Andes, including growing plants meant to improve growing conditions on the fields and to sustain family health. Thank you for your attention, and let's continue with the next presentation. Thank you to those who have placed their questions in the area, the Q&A. We remind you that you can continue writing your questions. These will be not responded in this segment, but in the following. Now we will give a space to the questions and the comments of the scientific community, and then we will have the participation of Juanfran López, who has a practical investigation study on the bioinputs in England, Spain, in diverse countries. We give you the space, Juan. Good. I speak from Spain. Good afternoon. It's a pleasure to share this moment with you, share my impressions with you. In my experience, that englobe the perceptions that has been carried out on the knowledges of the biols. But to emphasize likewise, we have the experience on the global scale. For me, one of the most important things that Brenda has mentioned very well is to understand the use of the ingredients. And for me, it's critical to understand the role of each ingredient, because this can help us to understand these ingredients. Then and the role that they have in this fermentation, we can recognize substitutes that we have locally. And then one of the other things that is very critical is a good quality of the ingredients that are utilized that are local and that you can have substitutes and they can be of good quality. Regarding the materials, this is the experience that I have in Africa. This is the adaptation because many times people do not use biofertilizers because they don't have the material, etc. So I have set several examples here to have an adaptations of the materials that we have locally. If we need for the bioferment to make an anaerobic instead of a valve, we utilize this is a bike pneumatic, or we make the fermentations, anaerobic fermentations, without having to acquire these blue drums that are the most recognized. And then it's very important to permit a fermentation, adequate fermentation process so that all that Brenda has mentioned can occur. In this metabolism of this microbiology that occurs during this anaerobic fermentation. Regarding the use, here that Brenda has shared to recognize the multiple uses that the biofertilizers use or the bioinputs or liquid from not just foliar inputs, but for land or it also could be for compost, et cetera, et cetera. And one of the critical things for me that likewise would be interesting is to see a good planning. Studies have been made, and if there's evidence that the use of biofertilizers or the liquid fertilizers depends on the cultivation or on the situation. If it's a doses with a product in the adequate frequency, there are many benefits or in key stages in the plant growth. There are studies that corroborate if you use a biofertilizer and use it 12 days before fertilization, you will be almost guaranteed, or there's a lot of probability that this plant will not have plagues or diseases when it bears its fruit. And then there's a misunderstanding that I think that Brenda also mentioned. For me, it's critical when we are planning with biofertilizers, et cetera, which is an organic focus. People then say, okay, herbicide, when can I do this? I think that this is to change the way that we think and the application of the biofertilizers. This slide for me is critical to understand the use of the biols and how to utilize them. And the fertilization is not just the revision of minerals, it's also the lesion of all these secondary metabolisms that Brenda has mentioned, the minerals and vitamins, etc. Then we go for a rescue agriculture, which is where we just focalize in concentrated microorganisms. This is a prevention cultivation. This is one of the benefits of the biol. So we have to understand this. With addition of a biol, then we have a resistant plant. So what we change in the focus or increase rendering of the cultivations through the increase of the plant health. So this is a different way of seeing it. The effects of the biofertilizers, principally, this is the secondary metabolites, then the fermentation process, the degradation due to microbiology that is present. Bioavailability of these elements is the quotation of the minerals and the colonization of this microbiology, for example, in the plant. I like this phrase that the countrymen are scientists, the farmers are scientists. We have seen in the revision, the biographic revision of the Andes, that there's many recipes, etc. So each farmer or in each situation, we have to take these processes or this use of bio to each area. there's a strategic photo with other tools that we can utilize. For example, I have put here a photo of seeds. And then many of the experiences that I have, many people try to make mini biols or many things that they try to apply. So we always recommend making one or two products, but that function or work correctly, and then understand the principles and modify the fermentations. And one thing that is important in perception, it's not a conjunction of recipes. The issue of producing or applying biols, it's a change of mentality. This is what I said in the system thinking. And to finish, some possible suggestions. We speak of not standardizing the production of biofertilizers, but maybe we could standardize and have some keys so that in a very easily understandable way and economical way, we can know this quality and processes of the biols. For example, here there are some analysis that are carried out with tools that are not very expensive. And here you guarantee if you have a quality biol, how the process is made, et cetera, et cetera. The pH will tell us if the fermentation process has been carried out properly or not. It will tell us if the final conductions of the biofertilizer are adequate. So we have the final product. It's an adequate final product. If, for example, there are charts that mix or that cross the pH with the reduced distillation, this will help tell us the functional group that predominates, or if there are pathogens, et cetera, regarding the biofertilizers. Regarding the content that I think that Brenda also mentioned, we can make an idea and have an idea on the content of complex molecules of these organics, vitamins, the bricks contact, and then through all of this, which are the total dissolved solids, we can see the content that of molecules, inorganic molecules, or simple molecules that, as I have seen, this can tell us in a very clear manner the application doses or the content. And then it is very interesting. What I want to comment is that a biol is not finished when fermentation finishes, but then after it has a maturation or stabilization phase, which is very complex. And to see it with a visual analysis, there's a tool, circular that I use it a lot. And here in the image, we see on the left a biofertilizer that is not matured and when the fermentation process has finalized. And after a time of stabilization and maturation at the right of the image, how this image tells us that this biofertilizer has matured or is stable. We can get deeper in, we can see the mineral content, organic, et cetera, et cetera. And then I have my colleague who will now present, we will delve deep. I would emphasize the importance of the applications in the critic points of growth. There are studies and evidences that say that what I commented before, determined doses in a determined time and in a determined manner make a lot of difference, impact on the plants. And then an idea simply, as Brenda has commented, we can have biofertilizers with vegetable remains. If we understand the principle in the biol and fermentation in the process, for example, then we can know we can utilize ferment of a resource. I'm setting an example. It could be sulfate, the fermentation. We will have a compendio of a product that have a lot of, this can be utilized for the agricultures with what I shared before, the agriculture of prevention. So reduce the use of copper that, even though it's here in Spain and Europe, it can be utilized in the organic culture, but it's very abusive. So to turn things around, the secondary beyond, and for me, the biofertilizers, in the hands of the farmers. Thank you very much. And I give the floor to Stephen. Thank you very much. Thank you to everyone. I will put on my presentation. Can you see it? Very well. Okay, here we go. Very good. Thank you, Juanfran. Thank you, Vanessa, Brenda. I think that you've been able to share a lot of the revision in the comments of Juanfran. They were very interesting. For me, from my point of view, my focus will be about the issue of that comes for what's happening in the future, some investigation tips that could delve deep into, as we have seen in the presentation, there are some things that to systematize better the effects or to distinguish the effect or to have principles in the design of biols, correct? I think that for me, there are two important days, and I think they go together with what Juanfran has mentioned. First of all, there's a very empiric way that goes in collaboration with the agricultures, analyzing their needs, what interests do they have. I think that there are questions in the chat regarding counting benefits and costs. What can cost, what is bio cost to make in view of other practices that the farmers have and they could acquire? What is the cost in terms of training? This is a very interesting and valid area of the investigation. Likewise, I think that for me, the empiric essays, there are some principles. One is to understand, according to literature and the existing work, what type of biol, or to classify the biols and to include other preparations, because biol, as we have seen in Brenda's figure and what Juanfran showed, they're just a small part of what there exists in the bio provisions, and to try to select these to express different hypotheses on their effects, for example, and the variations in pH, and then to see the impact in the plant physiologically and in terms of risk and stress. For me, it's fundamental. We can progress a lot in terms of this empiric experimentation if it's very well, if we have clear hypothesis. We have not found in the literature this evidence regarding ADAS, on the impact of response to ADAS, even though it has seen that there's foliar properties. Because we know that, for example, within the working group of the Andes, the McKnight, there are organizations have carried out these experiments, and we're in a process of trying to understand this and publish it. And it'd be very interesting to have a response here, because Juanfran has said that we don't want rescue material, but in this case, it's fundamental to recognize that it's important, it's vital to have this response to some risks and to understand if it really work and how they work. This would help us to improve. Another fundamental principle that we are supporting currently an investigation in Tanzania in the preparation of seeds with microorganisms, in homemade microorganisms with different components, and it's totally aerobic. It's not a fermentation, it's something else that maybe we should be learning more on the preparations of aerobic preparations. But the fundamental thing is to compare sterilized preparation through and different methods versus non-sterilized. What is the microbial contribution and what's a nutrient contribution? This is fundamental that should be practiced in all of the investigation on biol. And I have mentioned that there's a great variety of preparations, cultivations, and different incubations. We have seen and on my trip to Aruba, there's a universe of different preparations, microbes from the mountain, cultivating the microbes from different locations. And there are different protocols that would help us to understand these diverse inoculants that the agricultures are using. Let's go down. Okay, the second route is to understand what happens with the plants, the mechanisms. First, I would say that it's very important the empirical experimentation is very important to generate what we will be studying here, to not have a study on something that we know doesn't work. To utilize the literature with efficiency, to know previous experiences, not just in biol, but in other microbial issues or physiological issues of the plant. And for me, it's very important to distinguish what's functional, what's the functional change in the ground in terms of transformation of nutrients or prevention of plagues in the plants, to know the response in terms of its physiology, a highlighting of stress that can be influenced by this. And they're very complex studies, and we have to have an association with institutions, high-level institutions, that will have the methodologies to understand things deeper. Now, to mention this, some have mentioned trichoderm or syphilis, and we have the example of mycorrhizas. These are functional groups where a type of strong thrombosis has been demonstrated, whereas the strategy of biol is more associative. It's a more mix of things, and to distinguish these strategies, when we know that there's a correlation with the plant that is more important. When we have a biol or a preparation, it's more general. The impact that we will be, or even though there are specific things here, I mentioned the functional microbe. I will go to try to finish. For me, this was an interesting example because it's something related or similar to what we would like to see in biols. A very high-level study published in a high reputation organization has done a lot of work on one site to understand different inoculants. And the interesting thing is that they have had a positive witness, so to say, of product that exists in Brazil that is amply accepted and that influences on the rendering of corn. I think there's an agreement between agricultures and scientists that this exists. And the interesting thing there is, I set this as an example, a lot of work has been done to see if there are differences in the microbiome community. There are none. There's nothing that distinguishes these different inoculants. They're not biol, they're specific seeds of this. But what has been seen, and I won't explain all of this, it's an association between the components of rendering in blue and the different, not species, it's a not microbiome community, it's the functional aspects of the microbiome as calcium, solubilization of metals, production of salicylic acid, a type of coincidence that what we're talking about. This is what we find at the end, that the functional aspect, and maybe for me, this is a small lesson, that this work of delving deep can become very complex. And in this case, there's nothing bad of working empirically with good hypothesis and to sterilize, not sterilize, with other aspects, modifying recipes to see what this does, what the other does. And utilizing empiric block isn't bad because it's very complex. And what comes out of it is something that's very complex to understand. But each year that passes, these methods are more accessible. Genetics of land, of biome, of leaves, of the plant, and maybe that in this study in Tanzania, for example, we are utilizing simple tests to try to measure to see if there are differences in the microbiome community or not when the provision of seeds are utilized with the sorghum between sorghum with inoculants, which are truly a mix of different things similar to biol, manure, molasses, urine, other things that of course have different nutrients, but we want to see if they contribute to the microbiome. Studies such as these are very beneficial, especially when there's a collaboration with farmers and when they can receive and understand the results. I think that more than anything, this is my presentation according to the sheet, and this was my contribution. Any questions that you have? Welcome. Thank you for the work carried out, Brenda and Vanessa and Rikolto. And thank you, Juanfran, for your comments. Thank you. Thank you very much. Now we will go to the Q&A section. First of all, I want to say that we have gotten many questions. Some have been responded in written form. Those who cannot be responded on air or live will be responding in the next few days via email. And we will be sharing the presentation and the documents of the investigation so that you can continue learning and you can add these to your database. You can also send these materials with whom you think would also benefit from them. I will go over a few questions. I don't know if you want one by one or if I should give to everyone, and then we can go a round of questions. Okay, I will do it person by person. I ask you please to be brief. As I have said, we are a little bit behind time. I will start with Brendan. The questions asked, are there investigations that share effects of biols versus other taking advantage of the sources or organic material and nutrients, or with use of contribution in nitrogens, rotations, etc.? Good question. The greatest amount of studies that we found have comparisons between biols, and many times they did not compare these with other cultivation rotation methods. In few cases did we see comparisons between industrial fertilizers and bio. In this case, I'm thinking about a specific study. They controlled the amount of nitrogen that the industrial fertilizer, and they calculated the nitrogen in this bio and the compost. They were similar. And in this case, the bio increased the amount of the crop. But this is a very specific case, and I think that the point is obvious. There's a need to make these comparisons for the comparison itself, but also to see How can we deliver these methods that can be complementary? Thank you very much. Now the questions are for Stephen. Are you saying that the benefits of bio are contributors of minerals such as nitrogen and others? Can you see any difference between their direct use in the land or in the foliar part of the plants? Well, for me, it's an open question regarding what can happen in the land. And I think that this was the purpose of the revision. Just maybe we want to highlight that part of what the revision indicated, the hypothesis of the part of its effect has to do with minerals. This is very strong. This is what the fermentation does, is to liberate nutrients, among them nitrogen, what has been described in the revision. And then the part of the microbic area in this, I want to re-emphasize the question for my other panelists. I think that when they have said that they have not seen impacts on the microorganism life, they have found studies that they have investigated and have not found that the response was negative. It's not that they haven't found studies. That should be clarified with those who have made the revision. And likewise, in part for me, there are other ways of influencing the microbic land and occupants of the seed, different aspects, but something folial in the plant. For me, my hypothesis is that the less possible that would accept, due to the fact that we're talking about fermentation and the environment of the land is aerobic. It's a little bit different, even though there are impacts, and it would be good to continue investigating for me. I don't know. I hope this responds to the question. Maybe Brenda could add? Yes. Would you like to contribute? Yes, thank you very much. To complement this issue on liberation of nitrogen, I work in France. I worked with Rikolto. When we carried out this investigation, we worked a lot with partials. I will respond various questions at the same time because all are interconnected. We worked in fields where we had we cut a partial in two. On one side of the partial, the farmer had his itinerary, his normal and regular itinerary, and the other one, on the other side, he would use biofertilizers apart from everything else that he normally does. The only variation that we have is his use of biols, because many times the literature is not just the use of bio, it's not the only change. When we work like this, what we see in the analysis of savia, for example, is the increase sometimes in an incredible way of nitrogen that is contained in savia. When this doesn't have any comparison with the level of nitrogen contained in biofertilizers, it's because it stimulates the plant and it can stimulate its capacity to search for minerals or to search for nitrogen in the ground. What I want to say is that not necessarily do we have to apply nitrogen in the biofertilizer for the plant to suck up more nitrogen from the ground to be green. And there's sometimes with foliar application that's well done, with molybdenum, which is very small amounts that we need, we will be able to transform the nitrogen in the plant and for it to develop much better. And this plant can have an impact on the land. If we have a land coverage with green manure, we have this correct photosynthesis, but we apply a biofertilizer that has iron, the plant will have more photosynthesis and will inject more sugars to the ground. So the impacts are very different due to the types of ground, the nutritional state of the plant, et cetera, which makes it very difficult in a study to obtain generalities. That's why it's very interesting what Stephen and Juanfran and Brenda mentioned. We have to take into account the level of the ground, the level of the plant, and adapt the practices with the analysis and all of the tools that we have. This many times is lacking. We're lacking in the biographical studies that we saw. Thank you very much. The next question are for Juanfran. Is there any guide to identify microbiological groups with a pH redox with the function that comes in the middle pH? Yes, I answered I wrote out the answer. If you put, France has studies that for a few years ago has a lot to do with redox in the land, in the plant, etc. So he crosses these two amounts, and there are charts that, according to crossing these two values, the biol, pH, and redox, it tells you the functional groups of the microbiology. It tells you if there's pathogens, et cetera. So it's an indirect measure of knowing the microbiology that you have. Then if you want to have a microbiological analysis, I commented this in a meeting that we had previous to this one. I was told that about analysis, microanalysis, inoculate of microorganisms in the mountain that were through 80, and they've had 1,300, 1,400 different microbiology families. So if we go to the real food pH, it's very interesting, yes. A few seconds. This is an analysis of biofertilizers, and what we have to be careful in policies, etc., or by laws is the amount of heavy metals. This never overlaps with a biofertilizer. And then the other thing with the microbiology of pathogens, I think Brenda has mentioned, or Vanessa mentioned this, what we see, there will always be pathogens, but what we see is the amount that exists. We always, the E. coli and the salmonella, and that have a minimum of presence. So these are the standard amounts that we have to be careful with in the fertilizer, heavy metals, and these microbiology families. Thank you very much. Finally, a question for Vanessa. There's a cost of analysis and economic benefits for the use of biols. Thank you, Marcia. I think that the presentation of Stephen mentioned that this would be a line of information that's necessary to cover, taking into account the sustainability of the practices of the family farmers that are connected in different income they could receive and their entrance to the market. On the other hand, I wanted to take advantage of a question that can also generate confusion in some participants. Is that in the part of the presentation we mentioned that it would be important and interesting to standardize the formulation of bio for future studies. And then it was mentioned that it's beneficial for bio to be transversal. As these two proposals can be compatible, we require the versatility of bio in the context of biodiversity in the Andes to mention the difference in availability of manure, for example, of the Peru and Bolivia. On the one hand, we have that. On the other hand, to really know the effects of bio in controlled spaces, we need to have a standard formulation that with time can take to one or several formulations that can guarantee the effect or impact that we will have and to know how to monitor it. Thank you very much. As I said previously, we are making note of all the questions that we have not been able to respond in written manner, and we'll be responding these through email together with the presentations and the investigation document, as well as other documents to share the results. We have to speak about the conclusions and recommendations. In this case, we have a space of 5 minutes for each person, and we will utilize the same order of the questions. Let's begin with Brenda, then Stephen, then Juanfran, and finally Vanesa. Thank you, Marcia. A few points without any necessary priority, but some comments. I think that this issue of standardization and preserving the diversity, which is of a benefit of the youth diversity and diversity of resources in different places, this is key. But it's true that up to a certain point, we can do what Stevie and Juanfran mentioned, that there are ways of measuring both the biol properties, the density of the content or bricks, or the pH. But overall, the evidence in the field, in the ground, what we can measure regarding the land quality and the plants. So we can always standardize this. What does the plants and the land reveal? And it's worthwhile standardizing this process to have a tool to preparation maybe could help to incorporate the diversity of making bio in the different regions and different places. And then a comment about the microbes and the diversity. It's easy for me to say that my hands are tied and that I found no evidence or didn't see anything in the studies. And this doesn't mean that necessarily the communities don't change or they don't have an effect in the land when we use bio. We know due to many studies that it's difficult to move the microorganism communities in the ground, and we need repetition from other interventions, many practices to change the microbiome of the land. But what Renita says, this is very valid. There are phosphorus and there is a stimulation in the ground, even in the same microbiome that is present, that stimulates microbes to capture more nitrogen. Not necessarily does this mean that it stimulates setters of nitrogen. They stimulate the microbes that are in the ground to capture more nitrogen that is present in the atmosphere, for example. And it's very difficult to evaluate from what maybe happens in bio regarding microbes, and if they reach the land, and if they make any change or survive after the fermentation process. This will always be something difficult to evaluate. But what we didn't include in the revision is the part that several participants mentioned, which is, for example, the microbiome of the plant itself, what happens in the surface of the leaves with the application of biol. This is very fascinating to me, what happens in the land necessarily, because this can happen not so much in the long term, but during the growing period of the plant. And there's a lack of investigation. And this is key to understand the effect of bile in the plant. Thank you. I will continue. Thank you. Excellent information of Brendan at the end of what philosphere and the sphere of the, this is interesting likewise. And to investigate, it's not an area that I'm very familiar with. I would add likewise the biome that is close to the seed germinating and other types of bioinputs, and they're a very close area to the plant likewise could be an influential area that we don't understand very well. Just another observation. It's interesting to see in the chat, there's a lot of questions of doses, how much should be utilized, how should we dilute it, how many times should we use it, a gap of exchange of practical knowledge, and the example that they have asked Juanfran, where is this chart, where can they find with the pH and the redox, which the community microbiome. So it's interesting to think, and maybe this already exists, but to have a type of resource, common resources, or a website that utilizes these two inputs or sources of information. There is a lot of brochures, small books, websites that describe this, and I'm recommending this to carry out the revision of the literature to the future, focused on the practical things, and to synthesize or provide a resource. I don't think it's a job for this team, but maybe it's an interesting objective that could give us maybe a goal for the future. Thank you very much. These are my concluding remarks. I will continue. I think my travel is very short. I'll be very brief. Thank you, Stephen and Brendan. It's marvelous to hear and to share all of these things with you. As a conclusion, I want to emphasize that to obtain the benefits of a biome, for me, it's critical to understand what a bile is and to understand what happens in the fermentation process and to test the different options and to see in each case what works. But an understanding of how the bile works and what it's for, and many myths will be answered as we saw in the presentation. And thank you very much. A pleasure on my part. Can I go? Thank you. I just wanted to share my screen very briefly to thank you for everything that was done during this great meta-analysis, because I know that Brendan, Vanesa, Fiorella, who accompanies as well in this adventure, reviewed a lot of literature. And it was a great job because it had not been done before to synthesize this knowledge, scientific knowledge of white and gray material that had not been done before, of this meta-analysis that was done. Thank you very much for this great part. It gives us a great base to continue progressing. In very practical issues, we have spoken of different types of investigation that could be carried out what I would like to say is that we have to continue these experiments, empiric experiments, which are so necessary because it helps us to progress and make the scientific world progress. And the scientific world is progressing a lot due to what the farmers do in the field. Maybe we have to ask ourselves, who feeds off who more? Because it's a relationship that we'll say a symbiotic relationship, if I can use the play on words. And we shouldn't forget that if the health of the land is something that comes from the plants, the healthy soil is a healthy plant. They're very interconnected. And there's a lot of experiments and results that I have been able to attend. I just wanted to add to this. The health of the soil depends on the health of the plant. A plant in good, healthy condition is a very good, healthy soil. So when Stephen and Brendan talked about biosfera, and to understand how the plant works is the best and the most economic way of making healthy soil. And this is verified in the field with simple types of analysis. So we have to continue working on these issues and not just bet on the biofertilizers. This is one thing, but these are better yet when we add all of the tools of the agroecology to be able to have this unique health, so to say. And thank you very much to everyone. And I give the floor to Vanesa. Thank you very much, Reni. I would like to Juanfran on the part that has to do with the farmer, with the socio-ecological aspect. In this study, many perceptions of producers were collected, an issue that we won't delve deep in this presentation. You won't see in the material, in the written material. And so we would like to highlight how can we collect or confirm with these testimonials what we mentioned with Fran. There's a lot of difference between the producer and the use of the biol when they know the process of fermentation, with which who don't understand and they tend to desist from the practice when it doesn't work in. This is connected. We have seen with testimonies it works best from farmer to farmer, producer to producer, when they're connected to their community association or a cooperative. There are benefits beyond the productivity that the agricultural receives, and that due to the way of living in the different communities, these are valued by the agriculturists and their families, such as knowing that they're contributing to take care of their soil, recuperating the ecosystem, and eating in a more healthy way beyond what they can obtain as a monetary benefit. Finally, it's worth saying that this webinar and the study doesn't pretend to promote the use of bio with scientific backing or with repeated uses. We provide you all this information. We want all actors, not just the scientific community, the beneficiaries, those who continue with the project, investigation centers and all can give an application to this information. Thank you. Thank you to everyone for your conclusions and recommendations. Thank you to those who connected and who have been contributing to the chat and contributing with their questions. I reiterate that the materials will be sent via email in the next few days. And we give the closing words to Remi Thinard. Thank you very much. Thank you for your participation in this webinar. I would like to thank all of the organizations who permitted this great study that lasted for several years, where there were many exchanges and continue in the agroecology, there are thousands of routes. You can arrive to happiness and health of the soils and of the plants and of the individuals, and biofertilizers are surely part of this as well. So thank you very much to all. Thank you to everyone. Thank you to everyone. A pleasure. See you. Goodbye.